Welcome everyone to this Saturday presentation by the Science Circle. This week we are continuing a sub-series of presentations known as 2020 Revisited. So in 2020, the Science Circle was particularly active. We had a large number of presentations or panels that we conducted, and we thought it would be a great idea to just go back and revisit some of those with updates. So one of the nice things about this series is a reminder for everyone that science keeps progressing and understanding new things. So I will post in local chat the names and the YouTube URLs of the previous videos because we were recording almost everything in 2020. And so I'll just briefly say that we're going to have Phil Youngblood remind us about his Marking Time Consciousness, Cosmos, and Calendars talk. Then Mike Shaw will give updates in material science to update the recent developments in material science in 2020. And then I will give an update on a topic known as symbiogenesis, because my Darwin Day Heroes of Evolution talk was covering Lynn Margulis. So with that, I will step aside and let Phil take it away. Thank you all for joining us.
Okay, sounds good. I'm going to remain seated, and I see on the thing that Mike's supposed to go first. Do you want Mike to go first or me?
Your slide deck is already up, so you just go ahead.
Okay, I'll just okay, I'll move it a little closer here, but not too close. Okay, so anyway, time has always fascinated me, and I think others as well. And I did a presentation on time as it's seen by us personally, as well as how we measure it, and then how it's kind of abstracted into calendars. And so I've chosen a few, not Rosemary, but Percy said, yeah, okay. So anyway, from what I mean by consciousness is our ideas of time and space, and they vary from culture to culture and time to time. No pun intended, I guess. But anyway, for some of us, time may seem fixed, or it could be relative. For example, I remember a almost near accident where a car that was coming away from a robbery came right into my lane and was kind of playing chicken with me, and time completely stood still. At least to me, I mean, just slowed down really slow, and I could see exactly what they were doing, exactly what I was doing, and I got out of it by actually leaving the road. But it was very odd that everything slowed down. And now, as I get older, the days and months seem to go by. I really don't want them to be. And then also, is time a real thing, or is it just a construct? Is it just, as Aristotle believed, simply the measure of change, or is it something real? In Western culture in particular, and I'm using that term very lightly, is that we tend to think of linear, the arrow of time, progress, and whatever. Whereas there's a lot of cultures that think of it as a cyclical thing, including large scale where the Earth has been destroyed and then rebuilt several times, or the calendars are cyclical. And then we measure time by a number of different ways. By the way, there's a fun video, the one that I have there, and if I can chew gum and walk at the same time, so to speak, I'll get you that video there somewhere. Okay. That's the NIST one. Maybe I can find it here toward the end. So anyway, there are a lot of calendar systems worldwide. The reason why it's so green is a lot of people have adopted what we call the Gregorian calendar, the one that says it's January 31st, 2026. But there are a lot of calendars that have been used historically and that are still in use for different purposes, whether it's religious or political. Political, for example, the one in Japan that measures it from the year of the emperor, or the Hajiri, if I pronounce that correctly, or Hajri, uh, Islamic calendar that, yeah. And then there are indigenous calendars. There's other ones. There's one in Thailand, other ones, Mayan calendar, which I think is fascinating. So in other words, it's not just 2026 everywhere. It's just that people have adopted that one primarily for financial reasons and such. So if you look at some of the ones that are cyclical, you've got, this is probably one of the more known ones because it covers a lot of people, although it's not used. It's more used in ceremonial thing. In other words, the Chinese or calendar, which also could be called Vietnamese, or calendars. Yeah, you're a rat. So yeah, so that was just this last back in. Okay, in any case, the rat one was back in 2020, but I've updated these as we've been asked to do. I'm a snake. It is that the one this year coming up in February is going to be the horse. Now it's opposite the rat there, and it's a yang symbol, hot and light and other things having to do with that. Yeah, tiger, etc. And but this is a 60-year calendar based on Jupiter's orbital cycle. And you can see that there are elements of the week. There's 10 names in the week, not just because the week itself is not just 7 days. And then there's 12 earthly branches, and you can see that the cycle within a cycle there that then comes back to the same thing in 60 years. I find it very fascinating to study these and what's behind it. Yeah, exactly, horoscope, etc. And then there's another one, the Mayan calendar, for example, that is a 52-year cycle, where it's a cycle within a cycle. Yeah, water horse, or fire horse is another one. And then this one, instead of being based on Jupiter, is based on, yes, it's in other words, the Mayan and then the Aztec. Remember that the Aztecs came appreciably after the Mayans and adopted some of the same way that people come in and then they adopt some of their technology or beliefs. They adopted this calendar. And it's the 52-year one based not on Jupiter, but on Venus. Now, the Gregorian calendar, what's the Gregorian calendar based on? Yes, it's a base, a vesagesimal. I forget what the name of the base 20 means. Okay, what's a Gregorian based on? We all know. It's based on the sun and the moon, right? Mostly it's a solar and calendar, but you got months there too. But the big deal is the sun with 365 days, that sort of thing. So each of these calendars is based on in something a little different. Now, the Mayan calendar, as you might know, exactly, Gregorian, Pope Gregory changed it sometime back, 1562 or some other thing like that. Yeah, well, because they're almost the same with a few tweaks, like the year 2000, for example, which should have been a leap year, is not, because only leap years that can be divided by 400, only centuries that can be divided by 400 are leap years. So 1900 was a leap year, but 2000 is not, 2100 will not be. Yeah, vegesimal. Okay, and then yeah, well, yeah, okay, we can go into these. I could give you a whole hours of stuff, but the fact is that we think of time in a different way. And then if we start actually measuring time, if you get really precise about it, time is always relative, because we are influenced by gravity. And if you're influenced by gravity, as Einstein pointed out, general theory, is that under gravity, time is always relative to each of the people or things or whatever, because they each experience gravity in a little different way, and you've got all of these influences. So there is no such thing as a standard year, month, or even second. We have leap seconds because the day's actually slowing down, and there's all kinds of influences in how we measure time. If you get really precise and you look down at the bottom there, you can see that time, because of the gravitational influences, time is not just 365 days and change. It actually has a polynomial. I almost forgot the word. A polynomial equation that describes it because of all the influences. And so what we've had to do is, if we want to create things like GPS, then we've had to, or measure time very precisely for quantum computers and stuff, then we've had to not use, we've had to use things that are very small, like a cesium atom, that sort of thing like that, in order to have time be a little more constant. Yeah, well, we could go into that too, about what the difference between tropical and sidereal and that sort of thing. Okay, so in any case, now the ultimate clocks, though, and I'm watching my time here, the ultimate clocks are the clocks within our body, at least for individuals. And so you've got the circadian rhythms that can be messed up if you change your schedule or drink caffeine or ingest melatonin or other types of things. But did you know, for example, that or light exposure, that sort of thing. Light exposure is actually a good way to get back into sync with your circadian rhythms. But did you know about the blood pressure and temperature and reaction time, and when you should go jogging if you do that sort of thing, and all of and alertness at 10 o'clock in the morning? So all of that stuff like that. Yeah, daylight savings time can mess it up. And then finally, there's this one, which is, I find the strangest, and this is the last slide, is that you've got, and here people have been asking about this, but when you say life expectancy, there is no expectancy to be alive. It's just that what that means is the mean of how many people died before a certain date and how many people die after it, based on a particular peer group. And I'm trying to think, there's another name for the peer group. But for example, if you're born comparing, now this is in the US, which actually the cohort, perfect. This is for the US, which is actually, I think, 15th or something in the world right now. We've just, COVID really disrupted this. This was from before, this illustration was before the COVID pandemic, and we've just caught up to what it was back in 2015 in the US. And so, in other words, I got a couple more minutes. Okay, so anyway, the way you look at this as far as life expectancy is that, say, for example, you're born in either 1950, that's in green, or 2015. There's no expectancy to live to a particular date. It's just that if you take everybody, no matter what they do or don't do to their bodies or what their demographics are, if you take everybody, about 80% will live past 57, but if you're born in 2015, it's 69. The life expectancy, so to speak, for people born in 1950 is 68, and it's moved up to 79, which it just eked a tiny bit beyond that this last week or so. And then the greatest number of people will actually live beyond that by almost 10 years. And then some people, 20%, will live into their 80s and 90s. But if you outlast, I'm going to quit in about a minute. If you outlast people and say live past 70, which of course people in 2015 haven't done yet, But for the people who live in 2050 or 1950 time period, if you've already made it past 70, like me, that there's the average age that I could expect in theory would be to live to 86, provided I don't do anything stupid between now and then, or I don't, that sort of thing. So anybody? Well, yes, yes. And I have a couple relatives in the 90s and a couple that died in their 60s, so it's really bizarre sometimes. So, does anyone have any questions or comments? I see I'm no skydiving. Well, no, watch what you inject into yourself and what you do too. Yeah. Yeah, my time marking time is up. Or technically, we'll move to Mike in a couple minutes. But does anybody have any more comments or questions? I love to have them while we're doing this. Yeah. Anybody else on time? Go ahead. What?
Tagline is mentioning in the local chat that the shortest amount of theoretical time is Planck time.
Yeah, that's like 10 to the minus 43, I believe.
Yeah, he says minus 45.39 times 10 to the minus 44, and it's the time it takes for a photon of light to travel a distance of 1 Planck length. So that's an interesting,
like really small, like an atom or something.
Any other questions?
Okay, then we have time for Mike.
Okay, so I will bring my my slide deck down here and get it set up. Hopefully I don't delete the auditorium or anything like that. It's be dangerous to have me involved in anything these days. And let's see, I'm gonna move this guy, my Prussian blue. Or I'll just move that over there for now. Okay, so hopefully everyone can hear me. Let me know if you can't. I should be speaking. And I had some background noise issues, so I had made some adjustments to my microphone. So hopefully the background noise isn't so bad. All right. So, basically, we talked about, yeah, I don't have any cats around me right now, which is surprising given how many exist in the house. So basically, I had talked a little bit about material science in one of Baragon's Bistro episodes. So I kind of wanted to come back to it. And really, the elephant in the room on material sciences right now is the Nobel Prize that was recently awarded. Um, and before I talk about Nobel Prize, just some background. Materials, like what we use to do stuff. That's a fairly general statement, I imagine. But what we use to do stuff is really important. Um, we have, we as a species have gone through a Stone Age, a Bronze Age, an Iron Age, um, and arguably a plastics age. And then now we're looking at hybrid materials, hybrid very much on the molecular scale. And these hybrids almost get us back to materials that are very similar to stones. And I'll talk about that. So, Nobel Prize in chemistry was given for metal organic frameworks, or MOFs, to Richard Robson from University of Melbourne in Australia, Susumu Kitagawa from Kyoto University in Japan, and Omar Yagi at UC Berkeley. And these materials are important because they can be rationally designed and they have these large pores inside them where other activity can happen. And, you know, one of the things I did was uh, to prepare for this talk was kind of look at the YouTube, kind of look at the wiki page on metal organic frameworks. And I thought, yeah, that particular wiki page could do with a little bit of explanation. So I'm drawing heavily from there. So, well, and so my next slide here. Let's talk about materials. Um, as I said, we've got metal, we've got stone, we've got plastic. Um, each of those materials is yeah, each of those materials has like enough for a whole course of discussion. So I'm just going to talk about a couple of types of materials right now. Um, there's a material called zeolites. Um, Zeolites occur naturally as cat litter. You knew I would get cats into this talk a little somehow, right? Um, they're basically clay type minerals. Rationally designing clay type minerals is really, really difficult. There is a process called hydrothermal synthesis where you dissolve all the stuff up that you're going to turn into a ceramic essentially, and you cook it high temperature. High pressure and bleed off the water so you get slow crystallization. Um, Crystals are important for the study of materials, right? Because it is very difficult to characterize a solid. So if you have a crystal, that means that the atoms are arranged in a regularly repeating array in a regular pattern, right? Just like tiles on a floor. Um, and this regular repeating pattern allows for things like X-rays to be shot at a crystal and for us to get a well-defined diffraction pattern. Um, One can shoot X-rays at a crystal. Um, X-rays will be shot out. There will be bright spots of X-rays in a three-dimensional pattern around the crystal. Those are measured, crystals rotated, measured again, crystals rotated, measured again. That ends up being a data set. And with lots and lots and lots of math, one can take the diffraction pattern and work back to exactly how the atoms are arranged in a crystal. So this is like a definitive way of knowing what your crystal is made of. Really, it only works for ordered materials. It doesn't work for like, say glass, right? So like the borosilicate Pyrex dish that you have, it's X-ray crystallography is not going to work for that, right? So yeah, basically, looking over here, I have a laser pointer in this thing. There we go. Um, for some reason, whenever I use this particular um, slide um, um, slide generator, whenever I click the first time, my pointer turns sideways. So I hope everyone can see it. So what we're seeing here is structure of a zeolite. And zeolites are fairly old. And I said they're like very much like clays. And, if you zoom in into my slide, you can kind of see there's a, where the laser pointer is, there's a picture of a crystal. And um, over here on the left, what we're seeing is how the atoms are arranged in there. A zeolite is a aluminosilicate, so it really is a artificial clay, right? It's made of silicon atoms that are at the center of little tetrahedrons of oxygen atoms, and aluminums, which are at the center of little octahedra of oxygen atoms. These tetrahedra and octahedra are arranged in a way that builds up structures that have large channels in them. Right? So going down here, this bottom part represents a hydrothermal synthesis. So we have like soluble silicate, we have soluble forms of alumina, and they get cooked so that the individual tetrahedra and octahedra start to share vertices. Right. So down here, you can kind of see they start to share vertices and arrange themselves into these little, I don't know the name of this shape. It's not an icosahedron or something, but um, it's like a clipped icosahedron. And then these start to assemble into larger units, like this is a tetrahedral sort of unit of those icosahedrons. And I'm mentioning this. This is not a metal organic framework. There's nothing organic in this. I mention this, though, because it gives us the foundation for why people look at metal organic frameworks. With aluminosilicates, you kind of have to just take what nature is going to give you. It's hard to rationally design, right? Tag, I think you're right. Dodecahedron, it's got 12 sides. Each face is a regular pentagon. But then there's these vertices that have been snipped off here. So it's not quite the sort of solid that Plato would have liked. So what is a metal organic framework? Okay, so let's talk about Tinker Toys, because it is nice to rationally design a solid state structure. Uh, I would call this simple coordination chemistry. And, uh, back in the day when I was getting my PhD, my research group, we did organometallic chemistry. Everything that we did had metal atoms attached to carbon atoms. And that was a fundamental requirement for the type of chemistry we did. And we kind of poo-pooed the simple um, coordination chemistry. But it turns out that, since then, because this is like 30 years ago, there's been a resurgence in interest in coordination chemistry, especially for applications like these, right? So think about Tinker Toys. We've got rods, right? And the rods are things that look like this. And we've got a variety of different rods of different lengths. And then there are these linkers, which are these guys. And we could have a variety of different linkers as well. Right? I did a talk a long time ago, talked about Prussian blue. So let me trot that. Let me trot out a model of that. Boop, 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 boop. Where is that? So that's Prussian blue right here. There. That's Prussian blue right here. And you can see on the upper right-hand side of my slide, I've got a similar structure. Each vertex in the structure represents an iron atom, and it could be an iron 2 plus or an iron 3 plus. They alternate one after the other after the other. The rods, the linkers, are CNs. So CN minus is cyanide. So this thing is a polymer of iron cyanide, and some of the irons are in the 2 plus, some of the irons are in the 3 plus state. And essentially, Prussian blue is a pigment discovered 400 years ago, and it has this regularly repeating solid. The there's a lot of interest in this material as well because metal plus small linker, and technically it's a metal organic framework because cyanide with the carbon and nitrogen, a lot of people would call that inorganic, but it is the basis of a lot of organic chemistry. So what can you do if you take this philosophy of having Tinker Toys? Well, let me move this back out of the way. Click. And move to the next slide. So what if our linkers are things like little clusters of metal atoms? And if our rods are things like um, organic compounds that have functionality at two ends where they could attach to little metal complexes. So this is the image from the wiki page. On the bottom here is something that I've drawn. So the a, a classic example of a metal organic framework is made where your linkers are these little clusters that contain three metal atoms. And essentially, each metal atom is at the center of an octahedron. So I've drawn at the top of this little structure an octahedron, basically where all the oxygen atoms, I haven't drawn all the oxygen atoms, but all of the vertices are oxygen atoms. And um, in this fairly standard structure, what we have as these red lines are acetates, right? And you're familiar with acetate. It's what you get when you add, when you raise the pH of vinegar. So if you take this molecule over here, if you have a hydrogen attached to this oxygen, so it's a neutral OH, that would be vinegar. Acetate occurs naturally in salt and vinegar potato chips. Um, Acetate would be this guy. So there would be an O here, there would be an O here, there would be a methyl group sticking out somewhere in the middle. I've just drawn these as the linkers. Here's the thing. These acetates can be replaced. Notice how this O and the C and the double bond repeat in this thing that we're going to be using as a linker. Well, if one end of this linker replaces this loop, the other end can bond to another cluster. And that's what happens to take these kind of small starting clusters, and it starts to assemble them into super tetrahedra. So basically, one of these becomes connected to three others, and it forms a giant tetrahedron. And then these tetrahedra can be linked again, like in the zeolites, but on a much larger scale, to form these huge structures. In fact, one N. In fact, these larger structures are big enough to actually visualize in an electron microscope. So this is an electron micrograph of whatever this substance was, MIL 101. It's however the professors who created it decided to name it. What can happen inside these little holes is that each of these little holes can be a little test tube, right? And I'm kind of watching my time here. I actually wanted to go back, so let me go back. A feature of the chemistry here is that each of these chromiums, and you can have zinc, you can have other metals, has a water attached. That water can be replaced by something else. In the raw structure, those waters are intact. But what can happen after the raw structure has been created is that you can use these sites where the water can come off and be replaced by other molecules. So like carbon monoxide or nitrogen even, or hydrogen or some other gas. That would be a place where gas molecules could absorb. The amount of surface area that's inside these pores where gases and other molecules could absorb is just gigantic. And these sites, when you attach an organic molecule to a metal atom, you can cause a lot of things that are interesting, like catalysis, to happen. Okay, so yeah, so basically, this would be a example of what the structure might look like. I mean, those pores are big enough to see in a way. I mean, I know 10 nanometers scale here is that is a fairly large scale. But when you're a chemist and you say that your structures are 10 nanometers large, and you're really looking at like um, a tenth of a nanometer as being like where your normal molecular bonds are. Well, these things get big fast. So yeah, so these metal organic frameworks, the exact linkers you use, the exact rods you use, allow for a lot of control over a design. So here's a structure I just found yesterday, Thursday, or rather Thursday. I just browsed crystallography.net, had 5,000 hits for metal organic frameworks, and it's a small database. If you go to the Cambridge Crystallographic database, I bet you're in the hundreds of thousands there. This one had iron. Why, I like iron, so I just thought I would put iron um, into the search and narrow down. And what you can see, the side view, it's hard to see there. But if you look at the top view, this structure has a whole bunch of um, channels through it. And here's the thing, the it's got iron 2 plus in it, it's got iron 3 plus in it. Um, remember that hemoglobin likes to interact with O2. Iron 2 likes to interact with O2. If you are pushing a mixture of nitrogen and oxygen through this material, oxygen is going to get caught up and nitrogen is going to um, uh, bleed all the way through. Materials like this are the basis of how we actually separate nitrogen and oxygen these days. It's a much less energy intensive um, process than liquefying air and then distilling the nitrogen and oxygen liquids away from each other. Right. So, summary here. These things have so many uses and uses that have been realized, right? So as catalysts, replacing zeolites with these materials have allowed a lot of other types of useful reactions to be catalyzed. Won't talk too much about superacids and stuff. I want to make sure we have enough time for Stephen's talk. Um, the separators I talked about a little bit. And honestly, solid state electrolytes are very important as well. If we can make better lithium ion batteries or even get away from lithium as a charge movement thing, if we could just get to hydrogen ions, that would make lighter and cheaper batteries. Gas storage, low pressures. This is particularly important if we want to store hydrogen, because I'd rather not carry a tank of hydrogen around. The explosion hazard is terrible. Some of these materials will um, reversibly release hydrogen, and yet when they're full of hydrogen, simply be fairly inert. So that has a very good chance of being a hydrogen storage material, and for their mechanical properties as well. So I think at that point I'll stop. I do have another couple of slides, but I think I'm going to make them into a different presentation because they're a different aspect of material science. This has been um, an update on metal organic frameworks for which the Nobel Prize in 2025 was given. All righty. So I've been very negligent looking at the text here. Any quick questions? Do I have time?
Suma had a question about CO2.
About carbon dioxide. Let me scroll back up a little bit. Do, 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 do. having trouble finding. Do, 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 do.
Her question was just, what do they do with CO2?
Oh, okay. So that is an excellent that is an excellent question, really. Carbon. I mean, the interest in carbon dioxide right now is to take carbon dioxide and fix it, turn it into something more commercially useful and get it out of the atmosphere. So if we can have catalysts that will take CO2 in one of these pores and convert it, say, into methanol or something like that, or some sort of aldehyde, then that'll be very useful for getting CO2 out of the atmosphere. There's still a lot of bugs to be um, um, taken out of that, because just breaking down CO2 into something, into like methane or something like that, is a very energy intensive process. So one would tend to see such metal organic frameworks also coupled or tailored or designed to either be coupled to something on an electrode, or to have a photochemical sort of application, right? So it's like absorb a photon of light and then be able to do something like artificial photosynthesis to get rid of CO2 and make CH bonds. Yeah.
And Tagline had another question, and this should be the last one for the section. Do you see engineered catalysts replacing Nobel metals more over time?
Yes, absolutely. Because here's the thing, the noble metals are just getting more and more and more expensive. There's been a big push, especially recently, with getting rid of palladium in pharmaceutical chemistry. Palladium catalyzed coupling reactions, the Sharpless reaction, as it's known, is exceedingly useful, but the nanoscopic traces of palladium in the final products are exceedingly difficult to get rid of and can really be problematic. Palladium is in the same group as nickel, so if someone has a nickel allergy, you can probably think someone might develop palladium allergies. And the ways to get around this sort of thing is to have catalysts that are big, chunky rock things that don't leach, so metal organic frameworks are particularly useful there. There are also big pushes into catalysts that are 100% all organic and don't have any metals in them at all. So, yeah. So at this point, I will remove my screen and cede the floor.
All right. Thank you, Mike. So every year I give a talk on Darwin Day or nearby, uh, talking about someone whose theories, their work, their engagement with the community basically makes them a hero of evolution. And in this year, I talked about Lynn Margulis as well as Barbara McClintock. And these were two women who, again, made major impacts on our modern day synthesis understanding of evolutionary theory. So I wanted to honor Lynn Margulis again, because again, I've talked about retrotransposons and transposons before, but Lynn seemed very, very topical. So just a quick reminder about her. I'm not going to read this whole slide, but it's here for reference, is that, she was a scientist in zoology and genetics, and she got a PhD in genetics in California. And she was becoming her own scientist in a period of time where there was a lot of new understanding and a lot of debate with and about evolutionary theory. So she was a contemporary of Stephen Jay Gould, of Dawkins, and so a really exciting period of time, although still carrying a lot of this tradition of what we call neo-Darwinism, which had a lot of very strict ideas about how they thought genetics and evolution worked. So she was elected to the National Academy in 1983. And what is really important to recognize about her work is that she, more than many other people, highlighted from her research a fundamental redesign of how we understand evolution. So this is not a slide to read, but if you think of this as the too long didn't, what she is showing from her paper on the origin of mitosine cells is the idea that there are different domains of life, different types of species and organisms. And just as a reminder, there are very simple bacteria, and then we have larger complicated organisms like ourselves that are eukaryotes, that have very complicated and larger cells. And what she's showing in this diagram is the fact that she was providing evidence that some of these components inside the larger eukaryotic cells are actually, they used to be freestanding organisms that somehow took up residence inside a larger cell, and then they became a completely dependent component of the cell. They didn't have their own freestanding life cycle anymore, but then they actually contributed in some way to the biology of the larger organism. And so what I'll just mention, you've seen these from textbooks and whatnot. You guys are very familiar with the idea of mitochondria, which we call the power packs of the cell. They're what make energy. They do glycolysis. They're what allow eukaryotes to become big organisms, because the amount of energy you can get out of sugars is, many-fold more than if you didn't have this oxygen using machine. And then chloroplasts, we know, they're filled with chlorophyll, and these are this important conversion process of taking sunlight as well as inorganic carbon dioxide and then being able to convert that into chemically useful sugars. And this is really the basis of the vast majority of the biomass that we think of on the planet right now, is that there are photosynthetic organisms, in particular the multicellular plants, that are able to do this process, and then they create all the energy and food that organisms that cannot use photosynthesis basically get all their chemical energy from. So from that perspective, I want to say one of the really important things is that as we understand the vast majority of life on this planet, especially when it comes to the multicellular organisms, as well as things like protists and some of these other interesting organisms, that these were major developments that allowed life to be the way it is right now. And when we step back and think, what is the change to evolutionary theory that actually happened, is I'm showing from Darwin's notebook this classic tree of life, branching evolutionary pathways that describes his evolutionary theory. And what I'm depicting on the right is my own modification to that, which is sometimes new species actually come from the merging of other branches. And so two branches merge and in some way become a completely new organism, and then that leads to that whole set of diversity. And so, again, one of the things to really appreciate about Lynn Margulis from the time, and you can find essays about this, is a lot of her contemporaries were very dismissive of this idea. So it really took a long time for it to become considered textbook, although I think it was by the time I was in high school. And so she really had to fight through a lot of disdain for her work. And now I will say one thing that Lynn Margulis was trying to do, though, however, and this may have been a little bit of the issue, was that she was saying this explains almost all of evolutionary theory and diversity. And I think that's not true in terms of how often this occurs. Like I have depicted here on the right, how often is this a part of a major branch point of new species? I think she was overstating that case. But what's been really interesting since her work, and then even since I gave this Darwinian Day talk, we're actually finding lots and lots of examples of this. And so I'm just highlighting some headlines of this. I'll give you the quick upshot, is that the top example, a really interesting case where you have a a sea slug that seems to be capable of photosynthesis. And what has happened is that photosynthetic cyanobacteria have taken up residence. They had a mutualistic relationship, but then now they're finding that some of these genes that are from the bacteria are actually in the genome of the sea slug. And so this is highlighting a really key part of how these relationships develop, is that suddenly you have genes getting swapped between genomes. And so the big host organism now has the genes as compared to the organelle. So that was really interesting at the time. What I have on the bottom is another example that's more recent, where there are quite a large number of protists that are either mutualistic or parasites of larger multicellular organisms. And so C. ciona is a multicellular invertebrate, and residing in it is the C. intestinalis. And now what's actually interesting about the C. intestinalis, which is a protist, is that you find inside it an alpha proteobacterium, and it seems to just always be there, right? So what is the residence that's happening here? And what this paper showed is that this bacterium seemed to contribute the biosynthesis of lysine and lipoic acid to the protist. And so this is like a very early example of the beginning of the reduction and the participation of this type of endosymbiote relationship that then becomes potentially a completely new organism. Because as far as we can tell, the alpha proteobacteria, it's reduced a lot of its chromosome. So it's actually also taken in nutrients from its host. There are two more examples I want to give. And again, these really highlight to me how modern technology and modern sequencing capabilities really help us find and discover these types of relationships. And so the title here is that when you do some sequencing, again, you actually isolate a single cell, and then we have the capacity to sequence the DNA that you find within that single cell, is they were discovering, again, bacterial sequences within the eukaryote sequence. But what was really particularly interesting is that when they looked in termites for these bacteria and tried to stain them and scan them and look at what's going on, what they showed is that this bacteria will actually transfer from protist to another protist, even across different families. And so what we actually have here is not only do we have this idea of endosymbiosis that is or a potentially emerging symbiosis, but we actually also have this very in real time lateral transmission that is from one organism to a completely other organism of this microbe, which again could potentially lead to different ways these species work. And then the bottom one, and this is a really interesting one, is that when we think about large vats of fermenters and these cases where we create really high population densities, and you get some really unique new things that happen from an evolutionary point of view. And one of these concepts is something called quorum sensing. And what quorum sensing is that within a population of cells, you actually have a sense of knowing how many of you there are. And so it's kind of like a census. Basically, it's a chemical way of doing a census of the population. And this is important for, again, a lot of times how metabolic pathways are interacting from a low density to a high density situation. I think what was interesting in this case is what they showed is that there's this bacterium that's living inside the yeast. And what it's doing is contributing fresh chemistry. It's got its own chemistry going on that the yeast are able to sense for their quorum sensing, even though it's being generated by a subcellular organelle. And so this is a really interesting example of how the larger organism is taking advantage of the chemistry, not just for, like, say, metabolites or whatnot, but actually reincorporating it into how it understands the environment. And so, in summary, Lynn Margulis was very prescient in the fact that there's an amazing number of things that we need to kind of understand and tease out about relationships between different organisms that actually contribute to the whole. And while I think it's still overstated what she was saying about the actual speciation, because the threshold for speciation actually being kind of what she was describing is this exchange of genetic elements and say the reduction of genes from the dependent organelle into the larger organism. But these types of relationships that you see here definitely show that there's lots of examples of these relationships that lead to symbiogenesis that are really important for the current lifestyle of the organism. And so I think that is a pretty fascinating, and I'm really happy to be able to update what we've learned and what I've learned about Lynn Margulis. So with that, I'm happy to take any questions from a local chat, or raise your hand if you want to use voice. So Neuro Wonder mentions that Proteus mirabilis takes advantage of quorum sensing to find its way up a catheter to contribute to urinary tract infections. And yeah, that's a great example of if you're at a high density place here and you want to infect someone, you want to go to where you're a low density. And so, that's a great way to understand your environment. And yeah, bacteria, to some degree, quorum sensing is kind of an early way of thinking about how even things like bacteria that are not multicellular have this way to understand themselves as, say, a colony of individuals. Yeah, and Darwin mentions something, which I did mention in the original talk, but I didn't have time for now, is are there similar relationships between us and our microbiome? And so again, microbiomes, as a reminder, is this concept and idea that a lot of us multicellular organisms are actually a whole ecosystem, and that includes basically the bacteria, in some cases, multicellular organisms that live on us, and a lot of times we derive benefits. So the best example most people are familiar with is that our intestines actually help make vitamin K. So that helps us with blood clotting. The example I gave before with termites is a great example where this microbiome of termites helps them actually degrade the complex things like lignin and cellulose. And so, again, I think the microbiome, I think it's best to describe that as a separate relationship from symbiogenesis, because I'm not aware of anybody who's tracked the conversion of the residing genomes into our genome. And now part of this, of course, is because we, as multicellular organisms, we have gametes, right? So gametes are separate from the places where the microbiome is sitting. Our gametes, our sperm and our eggs, are a protected genetic environment in that way. And Tagline does mention, again, it's worthwhile to mention that the microbiome and its health has a major impact on your health. And so that's why, things like probiotics are considered a really important thing to keep in mind. Suno mentions, does quorum sensing play a role in the formation of biofilms? So biofilms are, again, the idea that bacteria find a chemical way to lock themselves to each other, kind of like chaining yourself in a way which is very helpful to resist antibiotics. To answer your specific question, does quorum sensing actually have some sort of signaling or chemical interplay with that? I don't know. I don't offhand think so, but I don't know. All right. Well, we're hitting 1 minute to go. I want to put this in local chat. So first of all, I'd like to thank Phil and Mike for contributing this update from 2020. I want to thank you all for coming and participating and asking questions. Again, I think